Thanks so much for the introduction. I'm excited to talk a little bit about my group at BMS and how we've been incorporating mass photometry into the AAV workflow in particular, but also a little bit about our workflow in general. So first I want to give a little bit of an introduction about what group I'm in and what we do. So I am part of the Discovery Biotherapeutics team, in particular Protein Sciences Characterization, or PSC. We're located in Lawrenceville, New Jersey, and we kind of categorize ourselves in 4 different quadrants. We do analytical and biophysical characterization of candidates, targets, and reagents. We also do protein design and engineering, antibody optimization, FC engineering, antigen design, that kind of thing. We're also studying early developability, looking at stability, aggregation, polyreactivity, liability risk assessment, and platform analytical fit. And finally, we're doing molecular interactions. We have a target or a lead candidate, and we have a and we want to study what the kinetics of binding are, the affinity. We do some high throughput screening. We do some mechanism of action of the candidate and the target, and so on and so forth. So a lot of our work is focused on antibodies, and not just antibodies, but different modalities, whether that be bispecifics, antibody-drug conjugates, Fabs, etc. We have a wide variety of techniques and instrumentation to kind of take a look at these things. And we've kind of broken it down here in, we take a look at, in the early stage, we're looking at antigens, reagents, tool molecules to help us discover our hits and leads. And in this, we usually are using intact mass, SDS page or CE SDS, ANSEC, studying freeze thaw and stability of samples, melting temperature, and initial SPR to determine binding. Once we get that stage and we're in lead discovery stage, we're still looking at antigens, reagents, tool molecules, and hits. We're using a lot of the same type of instrumentation, but we're adding in polyreactivity and aggregation studies to see, to study early developability and aggregation assessment. And then finally, when we look at lead optimization, we're looking at the same types of things, except with leads as well. We're looking at TM, TAG, using DSC to determine this. We're doing some viscosity measurements along with other groups within our company, and we're looking at mode of binding and in-depth analysis. So I have listed a lot of different techniques and instrumentation here, and what I'm going to talk about a little bit today is how we're utilizing the mass photometry within this huge span of instrumentation and how that's kind of fitting into our platform. So I'll give you a brief overview of how we're using the mass photometer in the antibody workspace before I move over to AAVs. So as I'm sure you've gathered, I'm going to be talking a little bit about AAVs today. In our group in particular, we're kind of mixed in between antibodies and AAVs. So what we've gotten is the 2MP instrument to kind of fit in between that versatility landscape. So when we're looking at antibody discovery, we're really being able to use the mass photometer in all 3 steps that I've outlined here: early stage, lead discovery, and lead optimization. Looking at initial reagent check, we're able to take a look at oligomerization, whether the reagents that we have or monomers, or if they're oligomerizing, aggregating, etc. We're able to check initial MAB hits and see if they're heterogeneous or homogeneous, and how this helps us with aggregation, stability, and early developability. This has been a huge help for us, especially when we're dealing with some difficult modalities that may give us problems on some other instruments that we have, such as analytical SEC or other things that could have problems with more difficult modalities. And finally, when we look at lead optimization, we can take a look at our lead MAB and our reagent and look at binding stoichiometry, which could give us a little bit of an idea of the mode of action and the mechanism of action, and how the antibodies that we're working at are actually engaging with the target. So finally, I'm going to take a look at this and our same aspect of what PSC group does at Bristol Myers Squibb. But in the past year or so, we've started incorporating gene therapy and early stage AAV production and QC into our workflow. And so the main question is, how can the mass photometer help us in that, and how have we been using it? So I want to give you just a brief workflow in Lawrenceville. So we're starting with AAV expression. A lot of the expressions that we're doing are expressions for biology work, animal studies. We're doing between 1 to 12 liter expressions depending on need. That will go down to purification, and usually in brief we're doing affinity columns, enrichment as needed, either mixed mode columns or cesium chloride gradients. Throughout the expression and purification process, we're doing viral titers. This is usually using a digital PCR to get us titer counts before and after purification. And then from there, we can take a look at detailed analytics, whether that be empty-full ratios, aggregation, assignment, etc. So I've kind of been the lead for our group in terms of analytics and that detailed characterization assessment that I was talking about. And so we had a couple different techniques that we already had in the lab to characterize full and empty of the particles that we're purifying. And so I kind of listed out what we had and the pros and cons of each. So the first would be dPCR coupled with ELISAs. The dPCR is really the gold standard DNA count, but if you couple that with a titer count from ELISAs, you get empty-full ratios. The problem with that is you're not going to get partials, and the combination of two measurements for empty-full ratio can lead to greater error, and we're already dealing with a process that could take a pretty long time. We could use biolayer interferometry or an octet system to use anti-AAV antibodies to quantify capsid titer. Couple this with ELISA, it's a little bit of an easier setup, and you couple this with dPCR for empty-full ratios. This is still something that we're working on and optimizing concentrations, but it's still a pretty tedious experiment, plus you're still going to be coupling with two different experiment types. A couple of the straight instrumentation that we were using for empty-full characterization is the Stunner from Unchained Labs. This is coupling absorbance with DLS and SLS for titer counts and empty-full ratio. This is nice when you're dealing with small volumes, and it gives you fast reads. It's plate-based format, so you could read about 96 wells in an hour. This is not a direct measurement, though, and you're not going to be getting partials. So this is something that we were having trouble with as well. And then finally, AUC, or analytical ultracentrifugation, sedimentation velocity. This is something that we considered as the gold standard for empty-full quantitation. But the problem with this is you're using very large sample size, and the quantitation can be ambiguous due to absorbance, even if you're reading at a 230 nanometer absorbance or if you're doing interference optics. This is also a very long run compared to a Stunner. And so when you take a look at the 2MP, how does this really compare? I'm going to show a little bit of some example data, but first I want to get into our measurement details and how we're usually doing this. We vary between either doing 2-fold dilutions directly onto the instrument or 10-fold dilutions. We're usually in the 3E10 or 4E11 CP per ml range. I usually don't worry too much about our VG per ml or actual gene of interest when I'm measuring. Mass standards that we're using are urease and apoferritin. I've had a lot of problems getting thyroglobulin. I don't know if anyone else has. And we also had a lot of problems getting apoferritin before. And before we got that, we were using BAM and urease. And that was actually working pretty well for us as well. We're usually collecting 1-minute movies. We're collecting at 2 to 3 different concentrations, and we average the full empty ratios. And we're usually using the 3,000 to 4,000 kilodalton mass range for empty particles, which could shift upon serotype. And then the full particle mass range depends on the gene of interest. So the first data set that I want to show you is taking a look at pre-cesium chloride gradient and post-cesium chloride gradient of an AAV5 sample with a 4,500 nucleotide gene of interest. And when we take a look at this sample, we're getting mostly empties when we're looking at our pre-cesium chloride. And after we do our cesium chloride enrichment, we're getting fulls in the 62 to 75% range at various different concentrations. And this was showing us really good data. So when we first started. But one of the questions that we had was, how does this compare to some of the other instruments, especially the ones that I outlined for you in the beginning? So when you take a look at this is an AAV sample that we had. It's AAV5 with the same 4,500 nucleotide gene of interest that we're interested in. We're looking at post-cesium chloride analysis and comparing that to analytical ultracentrifugation and stunner. And so one of the things that we've learned to do, I've heard a lot of talks in the other talks as well, is the mass photometer going to be something that you can replace with a different instrument, or is that something that you want to use as a complementary addition? And one of the things that we found really useful is taking all of the different instrumentation that we're having and using them side by side. And so in this case, you can see what we consider the gold standard of empty full quantitation being AUC, giving us about 9% empties, about 11.5% partials, and 71% full. But when we compare that to the mass photometer, we're getting very similar results, 69% full, 12.5% partial, and about 17.5% empty. And this is really impressive for us, especially with us straddling between the antibody and the AAV world. We're able to get these kinds of numbers using a 2MP instrument. In comparison, we're getting similar numbers for the percent full from a stunner, but the problem with this is we're not able to read partials. And so what's happening with the instrument is, because the partials are so much smaller and the extinction coefficient of the DNA is going to be different, it's going to be lumped in with your empties. So it looks like you have double the amount of empties than you actually have. And this is something that we've learned to live with, but it's really, since we've gotten the mass photometer, it's been really nice to be able to utilize both instrumentations, and especially when we don't have enough sample to run an AUC. The next thing I want to talk about is mixed mode purification as an alternative to cesium chloride. So what we were using for mixed mode purification is a Prima T beta column provided to us from Sartorius. And we provide a gradient, and we're able to get 3 peaks, peak A, peak B, and peak C. And you can tell that peak A is pretty much all empty AAV particles. Peak B is going to be mostly full with a little bit of partial, or not mostly full, but about 30 to 40% full with very little partials in there. And then peak C is going to have some partial species and maybe some overload species. When we take a look at this sample and compare it to the mass photometer, we're getting very similar results again. And this is also really helpful for us, because when we're doing these kinds of analyses and we're comparing methods and looking at something like mixed mode versus cesium chloride enrichment, we can see a big difference here. We're seeing a big jump in partials when we're doing cesium chloride as opposed to our mixed mode. And because right now we don't really know what to do with our partials, especially when we're quantifying it and using something like a stunner, you're not going to see it at all. And so it's good to keep eye of it, because we don't really know how that's going to play out for us down the road. And so taking a look at our partials and comparing it with these two methods, we're able to get really good comparison between the AUC and the mass photometer. And we're able to see that between a cesium chloride enrichment, we're getting about 20 to 25% partials. When we're doing mixed mode, we're getting about 10 to 5% partials. So a big difference in what we're looking at with our final product. One thing that I wanted to touch on a little bit is the resolution with the 2MP, especially with a smaller insert. In this case in particular, we're looking at an AAV serotype with a 1500 nucleotide gene of interest. And when we run this on AUC, we are able to see 2 distinct peaks that are different from our empties, and this would be a full as well as a doubly loaded full, or something that is being inserted twice. When we look at our mass photometer, we are getting the same types of percentages. We're looking at 8.2% full from the mass photometer, whereas we're getting about 9% total full from the AUC. But because of the resolution that we're dealing with the 2MP, we're not really able to distinguish between full single and full double. We might be able to draw some lines there, but it's really kind of guesswork to be able to say what percentages we're looking at. So we are a little limited in resolution when we're looking at the 2MP and dealing with AAVs. But in terms of what our group has needed it for and what we've been using it for, we've really been happy with the success that we've had and the types of resolution that we could see with these samples. So kind of going back to that slide where I'm comparing all of these different techniques, how does the 2MP kind of fit in there? This is really nice because it's a direct measurement of single molecules, which has been talked about before, but it really can't be overstated. It can determine partial species, unlike the stunner, and it uses small volumes and fast reads, and even though it is not plate-based and you're going to be, if you don't have an auto, you're going to be pipetting by hand, which we do not have an auto, we are pipetting by hand, but it's still a lot faster than dealing with an AUC. Partial resolution is a little limited, and the resolution is limited for smaller sized inserts, but from the data that we've been seeing, that can be fixed using Asamics. And we've also had a lot of luck with that with our system in general. So just to summarize, I want to thank the BMS team, especially the AAV team in Lawrenceville, but also the protein sciences characterization team that has helped me with not only the AAV work, but also the antibody work, and then our collaborators at different sites at BMS, but also the entire Refeyn team who helped us with our demo and helped us with a lot of troubleshooting after the demo as well. But please let me know if you have any questions. Yeah?
In your first set of AUC data, you showed by AUC you had about 9% other, but using the 2MP you had none. Do you have any speculation about why there's a difference?
That's a good question. That's something that we were kind of dealing with as we go along with our AUC work. One of the things that could be is I'm not really drawing out the overfulls to the larger extent. I don't think that's going to make up for the 8% difference. But I don't know if spinning is playing any role in this, or if we're missing some particles due to the methods of the mass photometer. So unfortunately, I'm not too sure. It's something that we've kind of been looking, as we've been doing these comparisons, we're looking for any types of differences that we can see throughout these. And that's why I wanted to emphasize as well that we are using these complementary and trying to take as much data analysis as we can and average them for a fuller picture, especially when we're working with a biology team who wants these samples for animal studies. We're usually giving them a table like this, even if when we're averaging the stunner, it's going to be inaccurate because there's not going to be partials in there. We want to tell them the full story of the different techniques that we use so they know what they're putting in their animals. Yes?
Very interesting. I had a question about the first part. So I would be interested, have you ever tried antibody-drug conjugates? And do you have any experience with that? How do they behave?
One of the things that I've tried with antibody-drug conjugates is just a general QC of the antibody-drug conjugates with a couple different drugs. And we were able to monitor at least 2 samples out of the 10 that we've tested that were more prone to aggregation than the others. And that's really what we were looking for. And so I believe it was from a different site at BMS that shipped us samples, and they were seeing trouble with their analytical SEC, and so they wanted for us to check the QC. And we were able to kind of identify which molecules to move forward with in their studies. And while this isn't ADCs, I've seen very similar stuff with bispecifics and other modalities where we're trying to run analytical SEC and we're getting very varied response, whether we're getting sticking to the silica columns or something. But as long as we don't have a large sample size, running on the mass photometer is usually our best bet to kind of deconvolute that messy data.
And if there are no more questions, thank you very much again. Thank you.